The information in our genomes is coded with chemical bases in DNA molecules. The entire human genome contains 3.2 billion bases, and each base is one of 4 types: A, T, G, or C. The order of these A's, T's, G's, and C's determines numerous aspects of the ways that our cells and bodies function. DNA sequencing is the way in which we determine the order of these bases. It is the method we use to study the human genome and discover errors in the genetic code called mutations. These mutations lead to many human diseases and play a pivotal role in the development of cancer. In pancreatic cancer genome research, our goal is to find mutations, errors in the DNA code, that are specific to the tumor and not present in the normal DNA. To do this, we use DNA samples from the patient's tumor tissue and the matching individual's normal tissue. Both genomes are sequenced and compared to each other to find mutations that are only in the tumor. Using this technique, we can locate cancer-specific mutations that might have contributed to the patient's pancreatic cancer. These cancer-specific mutations represent key targets for the development of new cancer screening and treatments. Here's a pair of DNA samples that we prepared from a pancreatic cancer patient. This one is prepared from excess tissue from surgically resected pancreatic tumors, while this one is prepared from the patient's normal blood cells. The samples are ready to go for sequencing. We've broken down the DNA into short fragments using a special sound wave, and we've added chemical tags onto the DNA fragments. Next, we make thousands of copies of each fragment for sequencing. To sequence the DNA, we add special chemicals one by one to react with the DNA, and then the sequencing machines detect specific colors produced by each base. The order of these colors gives us the order of the DNA bases. After analyzing the sample with different machines and computers, this is how we get the final sequencing result. Now that we have the sequencing results from the patient's cancer genome and normal genome, we can use a computer program to find DNA changes that are specific to the patient's cancer. We hope to use these changes to develop new strategies to diagnose and treat pancreatic cancer so that we can make a real difference for patients suffering from this devastating disease.